Hello everyone, welcome back to another class on regeneration biology. In today's class, we will learn about embryonic stem cells and how they are used and how different types of stem cells are there, which is in effect in human body, and how can we use them for our own benefit and for in regeneration biology, etc. So that is the overall theme of today's class. So in that, we need to understand first what is a stem cell and what identifies a stem cell from a different cell. So it is basically a cell that has the ability to continuously divide and differentiate, and differentiate basically means developing into a given cell type into different cell types, which can eventually become a tissue, which is a group of cells is called tissue performing a function, is a group of cells performing a task is called tissue, and how it can become different cell type. So in this picture there is a stem cell, and stem cell must have one feature to make a copy of itself by proliferation, which we refer to as self-renewal. So normally the self-renewal is a way of sustenance and propagation of the stem cell. Without this property, a stem cell, even if it is present, is not doing justice or not doing the adequate role to maintain a given tissue, or its contribution for sustenance of a tissue is minimal, and eventually it will become nil if stem cell population, no matter which stem cell we are discussing, if stem cell population is not self-renewed. And then stem cell must differentiate, not that a stem cell is constantly renewing and doing nothing. It is not going to help. It should have the ability to differentiate into any cell, provided if it is a perfect stem cell. Different types are there. Perfect stem cell, what I am referring to here is totipotent cell, and which later it is often referred to as pluripotent cell also, with a limited capacity. We'll see them one by one. So stem cells normally give rise to daughter stem cell, and this is the self maintenance, and then a specialized cell such as muscle cell, nerve cell, kidney cell, etc., it can differentiate, and this is the process of getting specialized. So stem cell has varying potential for when it comes to their differentiation, and one cell type is called totipotent. Toti is a representation of total. So totipotent can give rise to a complete individual, that is all cells in the body that is present. For example, cells from early embryo are often referred to as totipotent, means they are able to give an entire organism. That is how you end up getting monozygotic twins. Monozygotic twins are formed in a womb simply because this zygote during division it got separated. So it was a zygote cell which was a totipotent cell. It got separated into two pieces, and these two cells now develop into an embryo. If instead of two it is split into four pieces, you end up getting four identical twins. So this is what the concept of totipotency you should get. When you are referring to potency, it's basically the measure of how many types of specialized cell a stem cell can make. So toti means total. Any cell present in an organism, it is or it should be able to make it so that the entire organism is coming out of it. Pluripotent means one step below totipotency, can make all types of specialized cells in the body, but not a total organism. Say if you put a totipotent cell in a womb, in a uterus, it will give rise to a functional full-fledged organism. But if you put a pluripotent cell in the uterus or in the womb of a female, you will get a group of tissues, that may be teratoma, or a group of tissue means they are a combination of cell types that is formed and form a mass of tissue. Every tissue is formed, but it is not an organism. Then comes multipotent. So pluripotent cells examples are embryonic stem cells are normally pluripotent. They are taken from the inner cell mass of the blastula. When it comes to multipotent, it can make multiple types of specialized cells, but not all cell types. For example, tissue stem cell, say bone marrow stem cell, etc. They are multipotent. Tissue stem cell, it can give rise to, say, a given tissue has got 5 types of cells. So the stem cells of that tissue can give rise to all these 5, but it cannot give rise to another tissue type. Say, if I have got liver stem cell, it can give rise to different types of cells present in the liver. If I have got skin stem cell, it can make different cell types present in my skin, but it cannot give rise to blood cell. It cannot give rise to neuron. It cannot give rise to a nephron. So this is what you should understand about a multipotent cell. When it comes to unipotent, they are stem cells that can produce only one cell type, their own, but have a property of self-renewal as any other stem cell, which distinguishes from non-stem cell. Because non-stem cells, once they exited cell cycle, they normally don't divide. Fibroblasts are an exception. Fibroblasts divide, but they are not considered stem cell because they cannot differentiate into something. So muscle stem cell, for example, they cannot give rise to any other cell type. It can give rise to only muscle. So they are examples of unipotent cell. So what are the important characteristics of stem cell? One we already discussed, that is self-renewal. They are unspecialized cells because they are not committed for a function. Stem cells are not committed to perform a given function. They are unspecialized cells capable of renewing themselves through cell division, like any other cell will do, or like any fibroblast will do, sometimes after long periods of inactivity, because stem cells multiplication is demand based. Say I have 100 stem cells present in a given area, but none of them are needed because no other tissue type is dying. Like muscle stem cells, one example is there, that until and unless your muscle is damaged, what is the use of that stem cell? Your muscle can stay for long time. And if it is damaged, then say 10 muscles are damaged. Now I need to have 10 new stem cells come into picture. Then I have 100 cells as a stock. Then if I take 10 cells from that, then the stock will become 90, and you don't want that to happen. So 10 cells will divide and it become 20, and it becomes 110 cell now. 100, 90 cells kept quiet, 10 cells divided, and it became 110 cells. And those 10 will become muscle cell, and the stem cell population remained as 100 back again. So this is the principle in which they maintain. As and when it's in a simplistic sense, you can tell you are taking 1 glass of milk from a pot and you poured 1 glass back. Normally people pour 1 glass of water into it, but in any case the milk level will remain. It will get diluted, but here there is no concept of dilution. It will remain because a properly functioning stem cell is being kept. So why I gave this example is sometimes stem cell won't divide just for the sake of it. It will divide if there is a need for it. And regeneration, they can be induced to become tissue or organ specific cells with special functions. In some organs such as gut and bone marrow, stem cells regularly divide and repair and replace worn out, damaged tissues. In other organs, however, such as pancreas and heart, stem cells only divide under special conditions. Like I told you, in some tissues, tissue erosion is a given, say intestinal stem cell. We cannot imagine that, okay, my intestinal stem cell will stay for long time. No, because it is constant wear and tear is happening. Same your skin cells. Whether you walk, you run or do nothing, your skin cells are vulnerable for erosion because it's exposed outside. So it will erode and the stem cells will replace. Intestinal also, it is the same thing. But in some cells such as pancreas, heart, etc., the stem cell come into picture only if there is a damage. Now let us see what are the types of stem cell. One categorization we have seen: totipotent, pluripotent, multipotent, unipotent. Another categorization is based on where they are located, no matter which category they belong to. Embryonic stem cells come from 5 to 6 day old embryo, which is basically pluripotent. they have the ability to form virtually any type of cell found in the human body, but not a full-fledged human. For that, you need a totipotent cell. And embryonic germ cell, another group of stem cell, they are derived from a part of human embryo or fetus that will ultimately produce eggs or sperm, that is gametes. They are called gametogenic or germ cells, or germ stem cell, or embryonic stem germ cells, which basically mean they are present in an organism, but they give rise to only the eggs or sperm. So that is the purpose of this embryonic germ cells. Then comes adult stem cell. They are undifferentiated cells found among specialized or differentiated cells in a tissue or organ after birth. Say muscle cells have got stem cell, your retina has got stem cell, root progenitor cell, your skin has got stem cells, your liver has got some stem cell, intestine has got stem cells. So they appear to have more restricted ability to produce different cell types and self-renew. It doesn't mean that whenever a damage happens to that tissue, the stem cells are capable of fixing that damaged area. Not always, but there is a day-to-day manner. There are some cell death happen in a given tissue that will be taken care by the stem cell. But if there is a massive injury, a serious wound is there, then that adult stem cell are not powerful enough to fix that damage. They are meant only for running the day-to-day affair. They are not meant for fixing a damaged scenario. This is so in other animals which regenerate efficiently also, like say for example zebrafish. Zebrafish also have got adult stem cell, but they are not capable of fixing the serious damage to that organ. That is why differentiated cells present in that tissue have to reprogram and make a large number of stem cells so that they can be fixed. Adult stem cells are not able to run the show. And then comes the induced pluripotent stem cell, that is called iPS cells. And they are, like we discussed in the previous class also, Yamanaka factors. You take a fibroblast cell, they are adult cells of the body which can be reprogrammed to attain or acquire the pluripotency behavior. So based on the source, we have done this types of stem cell from where they belong to. So this is an egg, this is a sperm, and you end up making a zygote divided into 2, 4 morula stage, and you end up getting a blastula, and cells inside are called inner cell mass, and inner cell mass of the blastula, they are called embryonic stem cell, and that can give rise to the blastocyst. A very early stage of the embryo is called blastocyst, and then it has got outer layer of cells called trophectoderm. Trophectoderm will not become the animal, whereas the inner cell mass, which is called blastocyst, is the one which is giving rise to the animal. And then there is a full-fledged animal which grows to an adult animal, that is, in this case it is human. The fetus, baby, and throughout the life, these stem cells continue to be present in different part of the body as adult stem cell. Not that every differentiated tissue has got adult stem cell. Now let us understand a little bit more about embryonic stem cell, where we find them. One, we already know they are inner cell mass of the blastocyst, and they can be taken and decimated into individual isolated cell, and they can be taken from the inner cell mass and put onto a feeder layer, provide nutrition, and it will grow, culture, and you can differentiate them into all possible cell types or specialized cells. And embryonic stem cells, they are pluripotent in nature, and they are derived from the blastula stage, or they can be made from induced pluripotency, transfecting Yamanaka factors. So these cultured cells, when you allow them to differentiate, they can give rise to unipotent stem cells also. They can, not that stem cell always differentiate into, or the pluripotent cell or embryonic stem cell always differentiate into a tissue. Sometimes they can differentiate into another tissue specific stem cell also. Means it is less capable of giving rise to all cell types. So it is moved away from the pluripotency, but it is not differentiated also. So it is what you should understand, that circulatory system cell, nervous system cell, immune system cell, and they are a group of cells which can be seen in the early embryos also. They are capable of differentiating into a fully committed or fully differentiated cell, but they can have an intermediate stage also. So if you look more details about the blastocyst or the embryonic stem cell source, the blastocyst is a structure formed in the early development of the mammals, and it is the inner cell mass. In short form, it is called ICM, which subsequently forms the embryo. The outer layer of the blastocyst consists of cells collectively called trophoblast. This layer surrounds the inner cell mass and fluid-filled cavity known as blastocele. The trophoblast give rise to the placenta, that is attaching to the uterine wall. In humans, the blastocyst formation begins around 5 days after fertilization, when a fluid-filled cavity opens up in the morula, a ball consisting of a few dozens of cells, and that is what is used for the blastocyst formation, which is around 5 days post fertilization. The blastocyst has a diameter of 0.1 to 0.2 millimeter. 1 millimeter, how small we know? It is around 1 by 10 to 1 by 5th of a millimeter. It comprises around 200 to 300 cells following rapid cleavage, that is the cell division. After about 1 day, the blastocyst embeds itself into the endometrium, that is the uterine blood vasculature present in the uterine wall where it will undergo later developmental process including gastrulation, that is blastula mature into gastrula where 3 germ layers are formed. The inner cell mass of the blastocyst is a source of embryonic stem cell, usually used in culture. So if you look further, the characteristics, self-renewal is an undifferentiated state of long period. It can stay, and the maintenance of stemness, the so-called stemness or pluripotency has to be maintained, and this is a characteristic feature of the stem cell. And also it has the potential to form the teratoma when induced by in SCID mice, severe combined immunodeficient mice, which can the pluripotent cell can give rise to differentiate randomly and become a mass of tissue, a meatball which has got all tissues present in human body. Maintenance of normal karyotype is a necessary feature, that is the number of chromosomes, and clonality, ability to make a copy of itself, identical copy of itself. Stem cell markers expression such as nano OCT4 has to be constantly expressed in them, otherwise they can lose their stemness or the pluripotency. In clinical research, a lot of myocardium diseases, myocardium is the muscle of the heart, regeneration of damaged heart muscle by injecting human embryonic stem cell derived cardiomyocytes directly into the site of infarct. It's a published paper, you can read this citation. And also in lung disease, alveolar type 2 epithelial cell derived from human embryonic stem cells, and in a nude mice model, that is immunocompromised mice model of acute lung injury, that is also possible. And in nervous system, to fix the nervous system, oligodendrocyte progenitor cells derived from the human embryonic stem cells can improve functional locomotor behavior after cell implantation in damaged site. So 7 days after injury in the animal model. So these are all the different researches that has been done using human embryonic stem cell. Embryonic stem cell culture in the laboratory. Human embryonic stem cell are generated by transferring cells from the pre-implantation stage of the embryo into a plastic laboratory culture dish that contains the culture medium and also the feeder layer. The inner surface of the culture dish is coated with mouse embryonic skin cells and specially treated so that they will not divide. They will be alive, but they will not divide. This coating layer of cells is normally referred to as the feeder layer. And cells divide and spread over the surface of the dish. Some of you may be thinking why feeder layer is needed, because for stem cells to grow, it needs lots of growth factors. So when you feed them into this feeder layer and you inoculate them into feeder layer, these feeder layer will provide the nutrients, growth factors, etc. that will allow the stem cells to be stem cell and maintain as stem cells. So the plated cells divide and multiply and crowd the dish. Then they are removed gently and plated onto several fresh culture dishes. This process of replating and subculturing of the cells is often referred to as passage. So that is cell passage, it is often referred to as. Once the cell line is established, the original cells yields millions of embryonic stem cells that have proliferated in culture for a prolonged period of time without differentiating, and they are still pluripotent, are often referred to as embryonic stem cell line. It's a cell line. So importance of feeder layer, the mouse cells are in the bottom of the culture dish, provide the cells a sticky surface to which they can attach, and also the feeder cells release nutrients into the culture media. Disadvantages of feeder layer, there is always a risk of viruses and other macromolecules in the mouse cells that may transmitted into human cells, and future it can get into human. That is a challenge. So the differentiation of embryonic stem cell, blastocyst, fertilization, inner cell mass is collected, plated, and they are self-renewing, and you can make them into ectoderm cell, mesoderm cell, endoderm cell, germline cell, etc. You can differentiate them. So 3 germ layers that can be formed easily: ectoderm derived neuron, mesoderm derived blood cells, and endoderm derived liver cells. So the ectoderm gives rise to a lot of tissues that include spinal cord, nerve cells, hair, skin, teeth, sensory cells, eyes, ears, nose. So to fix all these troubles, for regeneration point of view, you can use stem cells that is initially pushed into ectoderm. Some of them are mesoderm derived cells, their muscle, blood vessel, connective tissues, and heart, etc. And endoderm derived cells, they are gut, pancreas, stomach, liver, lungs, urinary bladder, and germ cells, etc. come under endoderm derived cells. So the differentiation of embryonic stem cell, how do we differentiate? When removed from the factors that maintain them as stem cell, such as the feeder layer, ES cells will differentiate, and under appropriate condition, they can generate progeny consisting of derivatives of 3 embryonic germ layers, that is mesoderm, ectoderm, endoderm, and ectoderm. So general approaches of differentiation include: ES cells are allowed to aggregate and form 3-dimensional colonies known as embryoid bodies, or short form called as EBs. ES cells are cultured directly on stromal cells, and differentiation takes place in contact with these cells, another approach. It involves differentiating embryonic stem cells as a monolayer on an extracellular matrix protein. So there are different approaches through which you can allow them to differentiate. So ES differentiation, you have got undifferentiated ES cells. Initiate the differentiation, there is a leukemia inhibitory factor known as LIF, and embryonic feeders also there, and these are embryoid bodies, and these are formation of the incubating them with the stromal cells, and cells differentiated on an extracellular matrix. In all the 3 situations, they can give rise to different types of differentiated cells. So tissue stem cells or adult stem cells, this is what we should understand. Tissue stem cells, where we can find them, we have present in the eye, brain, skin, breast, testicles, intestine, muscles, bone marrow. All these tissues have got adult tissues, adult stem cells present in these adult tissues, and they will be running a homeostasis or day-to-day job. So adult stem cells include, they have unique characteristics. They are short form, they are known as ASCs. Adult stem cells, they have characteristics. They have the potential to self-renew for a long time. So we should understand, old people, aged people, they have wrinkled skin. Or you may see wrinkles doesn't come randomly. Especially you may have seen around your mouth. The people often refer to as laughing lines. Some people get foreheads, and some people get wrinkles here on the edges. So some places you get wrinkles, and if you think logically, they are the place where the skin is constantly put in stress. When you laugh, there is a fold that will be created. Another thing is you will get here, because whenever you smile, there is a folding happens on your cheek. So these are all the places where the damage is happening more than rest of the place. You often don't see a wrinkle here, or place where the chances of folding is less. So what happens during then? A skin cell gets damaged more frequently, and that will be replaced by stem cell. So what happens with old age? the damage is occurring at the same rate as it was in the young age. Slowly and slowly, your stem cell population in that area, not elsewhere, just beneath that damaged area. Stem cell is not, if it is a skin stem cell, it is present wherever there is a skin underneath. So the stem cells are depleted only in that place. It is not depleted here because cells were not dying here, hence I was not replacing. Here cells was dying more, hence I was replacing. But now the stem cell population is not as adequate, or because of age, although they are dividing, they are not dividing as fast as the damage. Hence you see a dent, you see a dip. Just like you may have seen on the road, people see a gutter, and in that gutter you put some soil, immediately it is sealed. After 2-3 times some vehicles go, that gutter will show like a ditch. So that kind of ditch that you see is called wrinkle lines. So they have the potential to renew for a long time, but long means not it's a given. So they can give rise to mature cell types that have characteristics, morphologies, and specialized functions along multiple lineages. Types of adult stem cell, they are hematopoietic stem cell because your RBC lives only for 4 months and they have to be constantly replaced. WBC, they all have got short lifespan, they have to be constantly replaced, and that's why in old age sometimes your RBC number decreases, WBC number decreases, because the same bone marrow is not producing as fast as it was doing in your young age. Mesenchymal stem cell, other stem cells such as neural stem cell, endothelial stem cell, intestinal stem cell, olfactory adult stem cell, mammary stem cell. And the sources location include bone marrow, umbilical cord, and cord blood. Cord blood is another thing which is present embryo to the mother, the connection cord blood. They have lots of stem cell because the umbilical cord is formed as a remnant of the embryo itself. So the cord blood cell have got lots of embryonic stem cell present. Adipose tissue, deciduous teeth, brain, peripheral blood, amniotic fluid, and amniotic membrane, synovial fluid, synovium, and placenta, they all have lot of stem cell. So if you see adult stem cell, this is bone, and inside the bone there is a bone matrix, bone marrow is present, and there are lot of different types of cell. So RBC have got its own lineage, there is a lymphoid lineage and a myeloid lineage of cells are there, and they give rise to different types of cell types that has produced. You can see hematopoietic stem cell, they give rise to multipotential stem cell that is able to give rise to lymphoid progenitor cell that give rise to natural killer cell, T lymphocytes, and B lymphocyte. They are called lymphoid progenitor give rise to. And then there is another called a myeloid progenitor. Myeloid progenitor give rise to neutrophil, basophil, eosinophil, and also give rise to the platelets and RBC, and also monocytes and macrophage cells. They are coming from the myeloid lineage. And this stromal stem cell present in the bone marrow, they can give rise to the bone or cartilage, and it can also give rise to other hematopoietic stem cell, and it can also give rise to an adipocyte, that is the fat storing cells. And some of this stem cells, they can give rise to the skeletal muscle stem cell and hepatocyte stem cell, that is coaxing them. Normally they don't produce. Can we take these bone marrow cells and you force them to make a skeletal muscle stem cell? Can we make them to hepatocyte stem cell? Technically it is possible through differentiation, and those kind of research help them to fix a damaged tissue. So this is a blood stem cell found in the bone marrow. They are multipotent cell. They usually differentiate into specialized types of blood cells, like I mentioned, myeloid and lymphoid lineage, and red blood cell, white blood cell, and platelets, which usually happens, but they can be forced to give rise to, as it is mentioned, skeletal muscle and hepatocyte or various other cell types if you give a proper atmosphere. So these are all the approaches that is done to make use of stem cell in regenerative medicine. So we should understand establishing these cells or isolating them and propagating them and implementing in the right place is the future of regenerative medicine. And we will study more in detail about other aspects of tissue regeneration in the next class. Thank you.